Hi, I'm Kavita, and today we are going to learn about two things in biochemistry and applied zoology. And that is, one is classification of DNA and structure of DNA. So let's say it's a chemical nature of DNA. The objective of today's class are to recognize the contributions of Chergaff, Rosalind Franklin, Maurice Wilkins, Watson and Crick, and that led to the uncovering of structure of DNA, explain the DNA structure, classify the types of DNA. Outcomes: explaining the components or the chemical nature of DNA, listing out different types of DNA, and explaining the structure of DNA, at least in basic part. Now, we all know that our hereditary material is DNA, and this DNA, it is a part of a chromosome. Just recall what you have studied in your lower classes, that our body is formed of cells, and cells contain nucleus and cell organelles. Nucleus is the major master controller of the cell, and it is because this nucleus has genetic material, which is wrapped in the form of chromosomes. Chromosome is actually a conjugated protein. Now, the DNA that is present there, it acts as a genetic material, we all know, because it transmits the information from one generation to another generation. This transfer of information helps in carrying out all the basic things that is specific to a particular species. Not only that, not just only passing the information from generation to generation, these DNAs as well as RNAs, in other words the nucleic acids, they also have the function of controlling the activities of the cell. That means they also help in cell functioning. When you talk about the eukaryotes and prokaryotes, I am so sure you are familiar with the term prokaryotes and eukaryotes. Prokaryotes are the organisms where the nucleus is not surrounded by nuclear membrane. That means the genetic material is present in the cytosol itself. Either DNA or RNA is present in the cytosol itself, cytoplasm itself. Whereas in case of eukaryotes, the genetic material is present in the nucleus, and the nucleus is surrounded by nuclear membrane. So that is the basic difference between prokaryotes and the eukaryotes. So in prokaryotes, these genetic materials, they are present in the cytoplasm, whereas in eukaryotes, they are present in the nucleus. Not only in nucleus, they are also present in the mitochondria. And the genetic material that we found in organisms, either in plant, animal, viruses, or even in case of bacteria, they can be of two types. One is DNA, the other one is RNA. DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. So these nucleic acids or genetic materials are macromolecules, and they are essential for life both in prokaryotes as well as in eukaryotes. So in this chapter and in coming chapters, we are going to discuss about these two types of nucleic acids as genetic materials as well as the master controllers of the cell. Now directly going to the types of DNA, these classification of DNA is based on where they are present. Are they present in the nucleus, or are they present in the other than nucleus? So in case of, as I said, in case of eukaryotes, they are present inside the nucleus, and in prokaryotes they are present in a portion called as a nucleoid. Because it's called as nucleoid because it lacks a nuclear membrane. And this DNA that is present in the nucleus or in the nucleoid, which is basically going to act as a genetic material of that particular organism, is called as a nuclear DNA. It's called nuclear DNA, and these nuclear DNA are generally they are linear and with the open ends. And these nuclear DNA are stable to some extent, since their mutation rate is very less. But there are always exceptions anyway. So these nuclear DNAs are generally stable, that's why they qualify as genetic material anyway. If it undergoes frequent mutations, that means in every generation you will have new characters. Okay, and that means in every generation you are going to see either a positive or an adverse character that might again get it will be exposed to all the variations of the environment, and survival might be difficult. So for a genetic material, it is important that it must be a stable one. So for generations, at least for number of generations, so that for number of generation you'll have uniform features. Okay, so nuclear DNA has that property that it has a comparatively less mutation rate. And based on location, you can also say that there are other types of DNA, non-nuclear DNAs. So for example, mitochondrial DNA, and these are present in mitochondria, that's why they are called as mitochondrial DNA or mtDNA. Okay, I am talking about animals anyway, and these are different from nuclear DNA because they are circular in nature. They are circular, they are closed. Okay, and in these mutation rate is much higher compared to the nuclear DNAs. There are other DNAs too, okay, the plasmids and all. So here we are concentrating only on animal DNA. So nuclear DNA and mitochondrial DNA are the two types of DNA that you find in case of animal cells. This is based on the location of the particular molecule. There are also different types of DNA, and these are based on the structure or the form. And the first type we are discussing here is A form. So this A form, it is a right-handed helix. See on this picture, you can, on this slide you can see a picture of a bDNA, the next type, but anyway this can be used for understanding right-handed helix. So right-handed helix means clockwise helix. The turn of the helix, you imagine a spiral staircase, buildings there are spiral staircases. If the turn is in the clockwise direction, then it is called as right-handed. If it is in the anti-clockwise direction, then it is called as a left-handed one. So right-handed and left-handed are the two types of coilings you find in these DNAs. So A form and B form are both right-handed helix. So that means here the coiling is clockwise direction. What is clockwise? See, in a clock, how the minute, the second and minute, these hands move, that direction is from like 12 to 1, 2, 3. So that direction is called as clockwise. So if imagine a clock where the minute and second hands are moving from 12 to 11, from 11 to 10 like this, that is called anti-clockwise direction. So if the coiling is in the clockwise direction, it is called right-handed helix. Both A and B forms that we have on slide, they are right-handed helix. Okay, and here for better understanding, we'll be seeing the structure of DNA in our next class, but just now let's have certain points. In this A form of DNA, there will be 11 base pairs per turn. See, one turn means whole 360-degree rotation. So in that one turn, how many base pairs will be present? 11 base pairs will be present in case of A form. Whereas the diameter of that molecule, okay, so diameter here refers to from this to this part. See, this one is a bDNA actually, but in case of A DNA, it is a 2.3 nanometer, okay, that is the diameter of the aDNA molecule, okay, or 23 angstroms you can say. And here bases are more tilted, okay, in relation to the axis. Molecule, if you take molecular arrangement, if you take the tilting, okay, sloping of these bases, it is more compared to the bDNA form. So these aDNA forms are generally found in experimental conditions. So they are specific at a particular like the humidity and all. So based on a particular humidity, you find these aDNA types. So usually the rotation, I said that the tilting, that is 32.7 degree, okay, right-handed tilting, and they are present in a condition of 75% relative humidity, and if the solution contains more sodium, potassium and cesium ions, in that condition aDNA is generally found. Okay. And the bDNA, the famous Watson-Crick model of DNA what we study, maybe from school to high schools and even college, which about which you are familiar, that is a B form, okay, based on its structure. So it is again a right-handed helix, and here the difference is that in aDNA it is 11 base pairs, whereas in B form it is 10 base pairs per turn. So in one turn there will be how many base pairs? 10 base pairs will be present, and the diameter of this bDNA, okay, that is 2 nanometer or 20 angstroms, 10 to the power minus 10. So here 10 to the power minus 9, the measurements are slightly different, units are different anyway. So either 2 nanometer you can say or 20 angstrom, that is the diameter, bDNA diameter of the molecule. And the helix, the length of each, each turn, the length of each turn is 34 angstroms. Okay, 34 angstroms or 3.4 nanometers. One turn of that helix is how long? It is 34 angstroms or 3.4 nanometers. So this is and even the pitch of the helix is 34 degree. Pitch of the helix is 34 degree, and it is found in 92% of the relative humidity, and when the ionic strength is low. Okay, ionic strength is very low. In that condition you are going to see the bDNA form. So almost all the DNA form that is present in the living cells, it is present in B form. Okay. So whenever we say the genetic material passing the information from one generation to another generation, we are referring to the B form and not to the A forms or any other forms that are present. The next type of DNA, a rare form of DNA that is present, is the Z form. It is different from both A and B type because this is left-handed helix. So here the coiling is in anti-clockwise direction. Okay, recall what I said about clockwise and anti-clockwise direction. So here the rotation is in anti-clockwise direction, and one more major difference between the bDNA and the zDNA, it gets its name as zDNA or zDNA you can say. Here the repeating units are dinucleotides. In bDNA it is a mononucleotide, here it is a dinucleotide, and there is also a difference that within the adjacent sugars face the same side, okay, they are towards the same direction in bDNA, whereas in zDNA they are in opposite direction. I am not talking about the opposite strands, I am talking about the adjacent ribosomes, sorry, ribosugars or deoxyribosugars that are present. And this arrangement, okay, as well as there is also repetition of GC bases. So this gives a zigzag movement, zigzag structure to the particular DNA molecule. That's why this is called as zDNA or zDNA, zigzag. Z is from zigzag. And here in each turn, recall in B form there were 10 base pairs. Okay, in A form there were 11, but in case of zDNA you find 12 base pairs per turn. So they are more compact. Okay, they are more compact, and the diameter is 1.8 nanometer. That means 18 angstroms. 20 and 23 angstroms in previous cases, but here it is 18 angstroms in case of Z form. And the twist of the angle, okay, is 60 degree. That means, see, it is what is this twist of the angle? It is like you imagine the staircase, the angle between first step and the second step, they are not at right angle to each other, isn't it? They are slightly twisted, and that twisting in case of zDNA is a 60 degree. Okay, twist of the angle is 60 degree. That means they are placed in such a way that from one step to another step, they are constantly at a 60 degree angle. Okay, step here I am referring to the base pairs that are present. So in each turn of the zDNA you find how many base pairs? 12 base pairs or 6 repeating units. 6 repeating units because they are dinucleotides. Okay, so that's about the A, B and D form. So this is how it appears in cross section. You can see how compactly it is arranged. Okay, A form, slightly irregular one, and then there is a B form, highly like a stable form that we find in most of the organisms, and then there is a Z form, the zigzag nature of it. Okay, zigzag, that's why it's called as a Z form anyway. And this zDNA, it is involved in, may be involved in gene regulation. Okay, it is involved in gene regulation, and presence of zDNA in natural conditions itself is an evidence that, I mean, at least it is, we are moving slightly away from, I mean, we thought that bDNA, I mean, the DNA is a stable one because it's a genetic material, it has to be stable. But at regular intervals, if you are finding zDNA, which is slightly different from bDNA, it can, I mean, help us to conclude that the DNA molecule as a genetic material may not be too stable. It shows some kind of changes, variations. It is slightly unstable one, because as you change the conditions, the bDNA might become a zDNA. So very light salt concentration will, I mean, that's the condition where you find the zDNA. Next, other than these three forms, there are also other forms of DNA that might be found in experimental conditions or in other adverse conditions, like cDNA, which is a right-handed form. And here there are 33 base pairs per turn, so highly compact one, and the diameter is around 19 angstroms, and tilt of the base is about 7.8 degree. And ddNA, that's another form. It has 8 base pairs, okay, per turn. See, 8 base pairs. Here it is 33 base pairs, and in another case it is 8 base pairs. Okay, and here axial raise is of 3.03 angstroms, and tilt is 16.7 from the axis of the helix, tilting. Tilting, recall I said how the arrangement takes place. There is also a eDNA, extended or eccentric DNA it is called, and this it has a deep major groove and shallow minor grooves are present, and it is a intermediate, okay, and found between from when bDNA changes to aDNA, the intermediate form is eDNA. These are rare forms of DNAs, but we have to understand that there are different DNA, not all the DNAs have similar structures. Now, moving on to the structure of the DNA, so we should be thankful to Watson and Crick, as well as Rosalind Franklin, as well as Wilkins. So, whose work have helped us to understand the structure of DNA. Now, this DNA, it is discovered by Frederick Miescher in 1869. Frederick Miescher was a student like you people, who at the age of 22, okay, identified, or he was working on certain things and was able to identify certain things, like in the he obtained actually isolated this particular molecule from the pus cells, okay, obtained from the discharged bandages, as well as he obtained it from the salmon sperm, and it was named as a nuclein. Okay, but he wasn't aware about the structure and the function of this nuclein. But anyway, he submitted his findings for publication. But editor was not sure whether, genuinity of this one, and it was held for 2 years, and then later on in 71 this article was published. Okay, but he continued his work even after the publication of that, and with all the experiments he was doing, he was aware that it is somehow related with the basic protein. The complex of basic protein with this nuclein was named as protamine. And later on many experiments were done, and the actual structure of DNA was identified. And this DNA, it is now known that it has a polynucleotide chain. Poly means many. When many nucleotides are polymerized, it gives rise to a DNA molecule. So when I say polynucleotide, So it means there are many nucleotides, isn't it? So what is a nucleotide? Each nucleotide is formed of a phosphate group and a nucleoside. Okay, phosphate group H3PO4 and there is a nucleoside. So nucleoside and phosphate group together it forms a nucleotide. Now the question is, what is this nucleoside? A nucleoside is nothing but a sugar plus a nitrogenous base. Okay, so this together forms, when it polymerizes, it becomes a polynucleotide chain. Confusing? Don't worry, we are going to see it in detail in next coming slides as well as in our next coming chapter. Now the sugar that is present in this nucleotide is a deoxyribose sugar, that is a pentose sugar, and there are different nitrogenous bases like purines and pyrimidines. First let's see the nucleoside. Okay, nucleoside is formed of sugar and the nitrogenous bases, and among nitrogenous bases we said there are two types. One is purine, the other one is pyrimidine. First see what are these purines and pyrimidines. Okay, purines and pyrimidines. Now purine, it is a double ringed structure. First, why it is called as a nitrogenous base? It is called as nitrogenous base because it has nitrogen. See, every molecule, the structure you are seeing here has a nitrogen in it. Since it has nitrogen and basic in nature, so it is called as nitrogenous base. And there are two types of such nitrogenous bases, two classes of nitrogenous bases, and this classification of nitrogenous base is based on the structure of or the number of rings they contain. So in case of purines, these are double ringed structure. So see here, they have two rings. Okay, two nitrogenous rings are present. If there are two rings, then it is purine. Whereas pyrimidines, they have single ring. If there is only one ring, then it is pyrimidine. If there are two rings, double rings, then it is purine. Record, these two, purines and pyrimidines, are types of nitrogenous bases which form a nucleoside. Okay, an example for purines, that means those which contain two rings, are adenine and guanine. See here, adenine and this is guanine. Both of them have how many rings? Two rings. And then we have thymine and cytosine, that is pyrimidine. There is another uracil that's found in RNA. So this is present in DNA. Two types of pyrimidines that are present in DNA are cytosine and thymine. Both of them have how many rings? Only one ring. Both of them, they contain nitrogen, so they are nitrogenous bases. Okay, so a nucleoside is formed of sugar and nitrogenous base. Nitrogenous bases are of two types, purines and pyrimidines, and this purine and pyrimidine is based on the number of rings they contain. If they have two rings, it is purine. If they have only one ring, then it is pyrimidine. Example for purine are adenine and guanine. Example for pyrimidine are thymine and cytosine. Okay, so that's about the nitrogenous base. Next one, the sugar that is present. Sugar here is a pentose sugar. Recall your carbohydrate class. So sugar, type of sugar, there are different types of monosaccharides depending upon the number of carbon atom present. If there are 5 carbon atoms, it is called as pentose sugar. 5 pentose sugar. Okay, and here the type of pentose sugar present is deoxyribose sugar. Now why it is called as a deoxyribose sugar? Because deoxy means one oxygen atom has been removed. Okay, where? From the second carbon atom. So what you have here on this slide is a pentose sugar because it has 5. See, first carbon, second, then third, then fourth, and this one is the fifth one, already numbered. So there are 5 carbon atoms, so it is a pentose sugar. This is called as a ribose sugar, but it is not truly a ribose since it has lost one oxygen atom, deoxy. One oxygen atom has been removed from the second carbon atom, so it is a deoxyribose sugar. So the sugar present in DNA is a pentose sugar. The name of that sugar is deoxyribose sugar, and it lacks one oxygen atom at second position. Okay, and this ribose sugar, deoxyribose sugar, when combines with the nitrogenous base, it becomes a nucleoside, as you can see here. Okay, and the one what we have here is a deoxyadenosine. Why the name deoxyadenosine? Deoxy here refers to the sugar. Okay, adenine is the nitrogenous base. Adenine is the nitrogenous base, then it is called as deoxyadenosine. If guanine is the nitrogenous base, then it is deoxyguanosine. And what about cytosine? Then it becomes deoxycytidine. And if the nitrogenous base present is thymine, then it is called deoxythymidine. Okay, thymidine. So depending upon the type of nitrogenous base present, you have different nucleosides. Okay, then the next one is a nucleotide. We saw nucleoside, that is sugar plus pentose sugar plus a nitrogenous base. When this nucleoside undergoes esterification with the phosphoric acid, that is the H3PO4, then it is called a nucleotide, and each nucleotide contains one phosphate group, then a deoxyribose sugar, then there is a nitrogenous base. Okay, nitrogenous base. Usually nitrogenous base is attached to the first carbon atom of the sugar. Okay, and phosphate group is attached to the fifth carbon atom of the sugar. So that means both phosphate group and nitrogenous base are attached to the sugar. Okay, so it is a pentose sugar backbone of the nucleotide. So each nucleotide is formed of one phosphate group, one sugar and one nitrogenous base. Again, depending upon the type of nitrogenous base present, we have different nucleotides, like deoxyadenylic acid. If it is a adenine is the nitrogenous base, which can also be called as deoxyadenosine monophosphate, because there is only one phosphate group that is attached. Okay, and if the nitrogenous base is guanine, then it becomes deoxyguanylic acid. Cytosine, then it is deoxycytidilic acid, and if it is thymine, thymidilic acid. Okay, so that is about the nucleotide. And the long chain of DNA, it is formed of repeating units of these nucleotides. Repeating, when I say it is not the same type, that's repeating. They are undergoing some kind of polymerization, unique way of polymerizing with the different nucleotides. Okay, so that's about the chemical nature of the DNA. We have not studied the structure of DNA. It is only the chemical nature of the DNA. So in this class we have seen the nucleic acid as a genetic material. Okay, and we also now know that the genetic information is passed through the in the form of DNA molecule itself. There are two types of DNA, nuclear and non-nuclear DNA, based on the location and based on the structure. There are A, B, Z or Z DNA, C DNA, E DNA and E DNA also. Okay, and each deoxyribonucleic acid or DNA is formed of two chains of polynucleotides. Each chain of polynucleotide is formed of nucleotides, and these nucleotides are formed of pentose sugar, phosphate group and nitrogenous bases. There are two classes of nitrogenous bases, purines and pyrimidines, and adenine and guanine are purines, and cytosine and thymine are pyrimidines, because they are single ringed structures. Okay, let's see how much you have understood. In a DNA, lack of oxygen is at first position, second position, fifth position or fourth position of sugar. What's your answer? Yes, it is in the second position of sugar. Okay, the next one, the nucleoside is composed of nitrogenous base and phosphate group. Nitrogenous base, phosphate group and sugar. Nitrogenous base and sugar, sugar and phosphate group. Your answer must be yes. Yeah, I'm giving time for you to try to read the question, options as well as answer. So what is the answer? Nitrogenous base plus sugar moiety is nucleoside. The bond present between two nitrogenous bases, hydrogen bond, ester bond, phosphodiester bond, pi bond. It is hydrogen bond. Okay. The left-handed form of DNA is aDNA, bDNA, cDNA, zDNA, and the answer is zDNA, left-handed. Rest all are right-handed. The purine present in DNA are adenine thymine, adenine cytosine, adenine guanine, adenine uracil. Purine means double ringed nitrogenous base, and here the purine present is or are adenine and guanine. Both of these are double ring structures. Okay, that is all for today's class. Thank you.